Okay, so my talk's on my work I do with natural toxins. So I run a natural toxins group, but we're out at Cooper's Plains in those wonderful new buildings, so we're not located here at St. Lucia. So we're going to talk about why indospicine is important among the natural toxins, some methods of indospicine analysis, and some of our recent work with camels. So natural toxins in agriculture. So toxins can be natural. People at Cooper's Plains, everyone likes having natural as being good for you. So I'm a bit of an odd one out. So Yasmin is in the audience now. She doesn't like it when I say natural can be bad. So natural toxins come from natural sources, including poisonous plants, fungi, cyanobacteria. So there's toxins in poisonous plants that affect grazing livestock. So there can be livestock losses, not actual death, just poor weight gain. There can be deaths, reproductive losses, but toxins can also form residues in meat products. So when we look at the pictures up there, a lot of people just say, nice animals, pretty pastures. I just say, there's poisonous plants. There's pyrrolidine alkaloids in both the fireweed and the Patterson's curse. Things that can poison the animals, they can leave residues in the meat. The cattle can actually do okay, but not very well. A lot of consequences. We also look at mycotoxins affecting health, human health and trade. That's fungal toxins in grain crops. A lot of those are regulated by legal limits. So in Africa, where we've done some work, they have the same limits, it's just nobody measures them, so it's an irrelevancy. So as I said, grazing livestock can consume a range of toxin poisonous plants across the northern Australia. Some of them are acute toxins, they cause sudden death. Those sort of things are easy to pick. If livestock eat something and die the next day, yes, you can work out what's poisonous. It's the cumulative toxins that are more insidious. And some of these things that stock have chronic exposure to. And concern was raised as to whether such toxins could form residues in meat tissues with possible human health and trade impacts. So when I first started in this work back in, it wasn't in 2000, but it was soon after, there was a SCARM report that raised down concerns for the industry. So what are the plants that we typically look at? The things with pyrrolidine alkaloids in them are the blue heliotropes, fireweeds, Patterson's curse up in the top. These all have a common core of pyrrolidine alkaloids, but they have lots of different esters. There's up to 300 or 400 of those sort of compounds. They all cause the same effect in livestock, which is chronic poisoning and liver disease. Indospicine, which we'll talk about later, and things like the bracken toxins, the tequilicides. Again, they are cumulative toxins, poison livestock, cause liver disease. It's rare that they'll poison livestock straight away, except perhaps in the cases where you introduce new stock to an area and they just go and eat it all. Mycotoxins are a huge class of compounds of 6 major classes, from the aflatoxins, the trichothecenes, the fumonisins, the rellanones, ochratoxins, ergot alkaloids. Lots of different compounds produced by fungi growing on different grains, different grasses. The ergot alkaloids down the bottom were always Barry's favourite. Okay, so as you can see, it's a diverse and complex range of chemical structures. We analyse toxins both in the plant tissues, but also in the animal tissues by LC-MS and GC-MS. So it's just a degree of scale. The plants always obviously have higher levels of toxins. The meat tends to have much lower levels. And then we've just had funding to get look at honey. It's you're looking at plants where the bees visit the plants and they transfer the pollen into honey, so you do get some of these compounds in the honey. So it's a whole complex thing, but the natural toxins that are out there in the pasture can actually affect us. So back when we started doing this research, when I joined DAF or rejoined DAF in 2003, we had a Meat and Livestock Australia project where we looked at assess the risk of occurrence of residues in meat from naturally occurring toxins to which ruminants are exposed across northern Australia. So we looked at all of those different regions, considered in total about 92 different types of groups of toxins. We looked at plants, at fungi, at cyanobacteria. We wrote some massive report of 265 pages, as you do with MLA. Okay, so we looked at the residue risk. We divided into a number of factors: the distribution and abundance of the plants, the consumption, the palatability, the livestock exposure, whether livestock are whether it's actually in the pastures where the livestock are, the toxin, whether the concentration and the toxicity, the residue persistence. A lot of toxins will poison animals but won't persist in their tissues. So if you're looking at residues, it's a different balance. Market type, live export versus slaughter. If you kill an animal, whatever toxins are in it at that time are sort of basically locked into its tissues. If you send it off on live export, they'll deplete over time. So we actually did consider that live export actually represented a lower risk as far as residues in meat. And after all of our 5 or so years, we concluded that in contrast to most other toxins, there is a likelihood of indospicine residues in livestock across all the regions up there. So that's across from southeast Queensland right across to the Kimberley and the Pilbara. So Indigofera plants that contain indospicine are distributed pretty well across that range. They are very palatable. The livestock are in the pastures where they are. It has high toxins. It is very persistent, which I'll show you later, and it's in cattle that are both going to live export and to slaughter. That's on a picture of 2 species of Indigofera, one of which, the one on the left, may be familiar to anyone who's a keen gardener because it's very prevalent around Brisbane, and the one on the right is Indigofera linnaei, which is more in the pasture regions. So it's a toxic amino acid. It's an analogue of arginine. It looks a very simple compound. It's found only in Indigofera plant species, of which there are more than 60 in Australia. Varying levels of indospicine, but the highest levels are in those 2 plants that are shown there. Indigofera are legumes, common legumes, more than 700 worldwide, predominantly in subtropical, tropical regions. So it's not just an Australian problem. It is a worldwide, well, a tropical, subtropical, worldwide regions. Palatable, high in protein, drought resistant. They were considered as potential fodder crops, which is when we get to spicata. Spicata is not a native Australian plant, despite its abundance in Brisbane. It was actually introduced. In fact, if you look at where the little red dots are where it currently is found up along the coast, the epicentre is actually Brisbane because that was where it was introduced, between Brisbane, Sanford and Gatton. There were 3 sites that they did trials on. They claimed that it wasn't ever released, but I think any gardener would tell them that's not really true. So it was introduced. It originated from Africa, Asia, and PNG, but it was introduced into Hawaii, Florida, Australia, Japan, New Caledonia, Micronesia. So everyone thought it was a good plant up until about the 1950s, when they started doing some toxicity studies. And the same toxins that are found in that are found in the native Indigofera linnaei. And while spicata only has a very limited distribution in Australia, linnaei, on the other hand, they're the herbarium records for it across Australia. It is a very common pasture plant. It's known as birdsville indigo. Widespread distribution across northern Australia, from the arid deserts of the central Australia to the tropical regions. Like when you can find it from Alice Springs to Bryan pastures, it's a pretty tough little beast. But in the arid regions, it can at times be a predominant plant because it has a long taproot. Again, anyone who's gardened with spicata and tried to pull that plant out of the ground will know it goes straight down. So the plant dies off during the winter, but when it comes for those first rains, that plant with the long taproot shoots up. So you get pasture that only has linnaei in it, not much else. Oh, that was a slide just to show that it's, again, not just an Australian plant. It does actually occur through Asia and the tropical regions up there. So the palatable legumes, high protein, intolerant to drought and salinity. But the problems came when they all started doing some of the testing. It was shown they had fed it to livestock for quite some time, until they started having some problems over long periods of time. And then they did feeding trials and showed it was a liver toxin, but that it causes abortion. It's embryo lethal and teratogenic. There's been abortion trials done primarily in Hawaii in the 50s, but later again in Fiji and Sri Lanka. There were some interesting trials done with rats and rabbits. It causes cleft palate in rats to an extent that if they fed it at a particular time in their life cycle, it's 100% cleft palate in those animals. Okay, as well as those effects, there's also a neurological condition associated with the Indigofera, but it's only seen in horses, not in ruminants. And surprisingly, there's actually 2 toxins in these wonderful Indigofera plants. There's indospicine that I've talked about, but there's also the neurotoxin, the 3-nitropropionic acid. Both toxins are actually present in both species of the Indigofera spicata and linnaei. So the neurological condition, it's not a cumulative toxin, but because it actually occurs, it's worth discussing. So it's attributed to the 3-nitropropionic acid. The horses, after 10 days of grazing the pound, they suffer from this progressive incoordination, dragging their legs in a pretty characteristic way, and if eating enough of it, they will actually die from the neurological condition. As I said, in some regions of Central Australia, you can have 25% of the pasture as Birdsville indigo. So that's normally when you get the horse disease. And we have actually seen similar neurological condition in horses feeding on pastures in Brisbane. So it's, the relationship between the 2 plants is really reinforced. But horses consuming Indigofera linnaei are not reported to suffer from the liver toxicity or abortive effects due to indospicine. But they do. But the interesting thing is when they are fatally affected by the neurological syndrome, they don't show the liver pathology, but they do accumulate the indospicine in their tissues. And they accumulate it to the point where meat from those horses will actually kill dogs, which is the topic of the talk. Indospicine. It's an arginine analogue, non-protogenic, occurs as a free amino acid. It doesn't join with other amino acids and form protein as arginine would. So it's experimentally shown to cause liver degeneration, teratogenic and embryo-lethal effects. It acts as an arginine antagonist. It interferes with arginase, but it can also interfere with a range of metabolic processes, because arginine is not just used for protein. It's a precursor for lots of things like nitrous oxide, urea, polyamines, so it has some of the effects of indospicine aren't actually well known because it can actually just interfere with such a wide range of cycles. But accumulation in animal tissues is interesting, and it's quite unusual to the extent that indospicine accumulates. So it's not incorporated in protein, but it accumulates as a free amino acid. We've carried out some feeding trials, but other people have carried out with other animals as well. So there's been trials with horses, with cattle, with rabbits, with goats, and now our camel workers, which we'll talk about. So all of the animal species demonstrate a similar accumulation of persistent indospicine residues in muscle and serum. And the notable thing is that these residues are actually still detectable several months after you stop feeding the animal the indospicine. So it's not only that it accumulates, but it persists. So it seems likely that all animal species consuming Indigofera species accumulate indospicine in their tissues. Every animal that's been tried, it accumulates in. Even in the dogs, it accumulates in their tissues. However, there's different animal susceptibility to the toxicity. So some of the animals can accumulate the indospicine in their tissues and only suffer really quite mild liver effects. On the other hand, our favourite dogs can actually be poisoned by the meat. So this is called secondary or relay poisoning. So the sensitivity, horses are right at the least sensitive end, with dogs in the middle. We have dogs at the far end. In the middle is the rats, the mice, the rabbits, the cattle, the sheep. Humans, we believe, fit in that middle class. We have done some trials with liver cells that give us some confidence of that, which is good because dogs are normally seen as a model for human toxicity. So that's some of the data about what the cattle fed at 5 milligrams of indospicine per kilo had no apparent effects. Horses fed at even higher, had only mild liver. Dogs, on the other hand, fed at only 0.13, so it's about a 50th the level, produced mild degenerative liver changes. So the first classes of the secondary poisoning occurred back in about 1984, and there were 30 dogs died in Alice Springs from feeding on horse meat. It was shown to be indospicine in the tissues, and it was demonstrated that horses fed the Indigofera linnaei accumulated the indospicine in their tissues. So some of the data on the amount that they were fed over so much time, they produced meat that contained 16 milligrams per kilo of indospicine. And that meat was then fed to 2 dogs for 32 days with all the normal elevated dog liver enzyme liver damage. So given the levels, you're feeding at different levels, but you're introducing more toxicity in the second animal. So back when we were doing our projects in 2009, 4 dogs died from severe liver disease in Perth after consuming a commercial Australian camel meat, sweet potato diet. These were just domestic dogs. They were in suburban Perth. They were actually being treated for skin allergies, which is why they were on this strange camel meat sweet potato diet as a non-protein diet. So they were actually typically pedigree animals. People were not happy when they died. Okay, because they were being treated for something, they were actually been consuming the camel meat diet exclusively for between 30 and 139 days. So unlike most pets where you feed them different things every day, or we hope people do, these dogs were actually being fed repetitively, just as you would a feeding trial, but Lex, we didn't need animal ethics for this. So all the 4 dogs that showed up showed inappetence, elevated liver enzymes. 2 of the dogs didn't respond to support and were euthanized. We did the indospicine analysis in the camel meat and led to the conclusion that camels were consuming some Indigofera plant. And surprisingly, someone has actually done studies of the preferred feeding plants of camels in Central Australia, and Indigofera's up there. So those are some of the levels we saw in the muscles, and they were the levels we were measuring in the dog skeletal muscle level. We did actually get some of the diet to analyse as well. Having found the 4 dogs that were actually sick, they then thought, maybe we should check the remaining dogs that are on that diet. So there were a total of 19 dogs. The top 4 are the ones that were actually clinically sick. The days on the diet, so they were on varying levels. We were never sure that the diet was actually all consistent all the way through, because some of the dogs seemed to be on it for inordinately long levels but only had very low levels of indospicine. So of the 19 dogs, 1 to 7 had elevated liver enzymes. The interesting one is dog 5. He was asymptomatic at the time when we were doing the testing, and they thought he was going to be okay, but 5 months later he developed liver toxicity and had to be euthanized. So all of the other dogs had no ill effects. People always ask, where are the camels in Australia? Because they're not in coastal Queensland, so we don't see them. There's a large populations of camels through all of the sort of arid regions. These are feral or introduced wild animals. They were introduced into Central Australia by the Afghans when they were transporting things across the middle before we had motorised transport. They were let loose after when they were no longer required. The current population's about 300,000. There were various estimates that there were 1 million plus when they started the camel cull, which interestingly gave us samples for analysis. There's generally about 5,000 per year that are harvested for human and pet food. It could actually be higher, and in some ways the finding of the indospicine in the camel meat has actually contributed to sort of the decrease in the use in the pet food. So what we actually, when we started looking at the camel meat, one of the things we actually really wanted to do was, because this one case had stopped an industry, which the Aboriginal population in Central Australia were quite keen on developing, we wanted to know, really, were all camels bad, or was it just the 1 camel or 2 camels that they'd used to make that particular batch of pet food? So yes, I think, yeah, it was known that the camels were consuming the Indigofera because there was been large studies done particularly in the Aboriginal regions about what the preferred feeds were. So the level of indospicine, Indigofera linnaei, is up to 2,000 milligrams per kilo, or a gram per kilo, which is highest in the new spring growth. So there's a lot of indospicine in the new growth. What I really was talking about, some of our analysis methods. So when we first started, we were back with everyone else. We started doing amino acid HPLC analysis. You can derivatize indospicine like any other amino acid, but you get these profiles that are really messy, and you're trying to find an amino acid among other amino acids and detect them by UV or fluorescence, which is not a very definitive method of detection. So we moved on to LC-MS as soon as we had the instrumentation. So with LC-MS, you get breakdown of the indospicine, like the molecular weight of indospicine is 174, and it gives you characteristic ions, whereas things even like as similar as arginine, which is only 175, has different breakdown. So you get different patterns. You can actually tune the machine to look not only for the molecular ion, but its transitions. So we quantitate on indospicine on the transition of 174 to 110. It's unique to indospicine, highly specific detection methods. So instead of those really messy traces, we just get these traces with single peaks. So our detection limit's really quite low. That's actually spiked muscle. So that you don't see the muscle background, you just see the indospicine. So because we were intending to analyse an awful lot of samples, we were looking for a method that we could analyse quickly, repetitively, set the machine to just injector after injector. So we look at underivatized indospicine. We just do a simple extract into heptafluorobutyric acid. It keeps the indospicine on the column a bit longer, gives us a bit better retention. We just filter it and inject it into the LC-MS. We have a retention time of about 5.9 minutes, run times at 10 minutes a sample. Auto-injectors are wonderful things. You can set them to go overnight, over the weekend if you try. One thing we did find was the matrix effect. While we couldn't see the matrix actually affects the signal. So that we were spiking and getting recoveries that were okay, but they're not brilliant. They're between 65 and 114. So it was the ones that we had matrices, in some of the samples was suppressing the signal, and other samples it's actually increasing the signal. And our dilution experiments diluted this, showed that this was primarily a matrix effect. So it's due to the muscle and the other tissue. We don't normally see it in the plant because we actually dilute those to such an extent. But in the muscle, where you're actually trying to find really low levels, you've got a fair bit of matrix behind it. So we did. Wanted to do stable isotope dilution assays using isotopically labelled internal standards. And James, who's here, who's going to answer any questions anyone has about the synthesis, synthesised D3 indospicine for us for this purpose. So this is it's only been accepted. It's actually been published now. So the synthesis of indospicine starting from commercial products, but you introduce the deuterium along the way, so you end up with indospicine that just carries a couple of deuteriums as a label. It has a very similar mass spec to indospicine, just that everything's displaced by 1 or 2 units. So chromatically, the deuterated indospicine elutes with indospicine. So the same matrix effect. If the signal gets suppressed, the deuterated one gets suppressed as well. So you do it by a ratio. You can do the quantitation by a ratio on the two transitions. So you can see the indospicine's one of the bigger peaks, the deuterated indospicine, but that ratio will always be the same no matter what matrix you're in. So our recoveries are now much closer to 100 and also very consistent across all tissues. So some of the samples there, camel meat that we actually got from the Simpson Desert, blank meat and spiked meat, they're all just sitting, very nicely, very consistent. I had a wonderful PhD student, he's gone now, but he did loads of showing it intraday repeatability, reproducibility, all the things that analytical chemists love, that it was really just spot on. So once we had the internal standard, it enabled us to do an awful lot more analysis than what we had done before. So we've done the plant analysis across loads, across the species, seasonal effects, camel feeding trials, the camel meat analysis, and degradation studies. So we looked at as many of the Indigofera species as we could get hold of. So the 15 most commonly available ones we looked at across Australia. Indigofera linnaei and spicata were the only ones that had reasonable levels. And even within those, there was quite a bit of variation. Most of the variations seemed to be on a seasonal basis. That histogram is across a number of sites where we collected Indigofera at different times. So in the early new growth, the September, November ones, the values are much higher than the same plant material collected later at the same sites. Remembering this is in arid regions of Central Australia. That's what the pasture actually looks like. So in November, that was the site, and we have these little Indigofera plants that are coming up. So you can see why the animals would be eating that when there's not much else there. Whereas in February at the same GPS location, you're looking at a fair bit of green material, and you've got bigger plants, but it's not only is that has a lower level of indospicine in it, but it's a much less part of the pasture. So the risk of indospicine intake by livestock is greatest in spring when the indospicine concentrations are highest and when it represents a much larger portion of the available forage. Not surprisingly, this parallels the incidence of Birdsville horse disease, the neurological condition. So the horses similarly get exposed to pastures that look like that, and that's when they get affected as well. So we did a camel feeding trial to look at where it accumulates in the animals, which tissues, and whether or not the camels were actually affected by it. We fed them at known rates of Indigofera spicata, containing a known amount of indospicine. We biopsied them. We had 3 camels that were euthanized after 33 days, and the remaining 3 camels were fed on the indospicine-free diet for a further 100 days. And again, we used our quantitation method for plasma, muscle, and necropsy samples. So this is Eddie, who did all this work. He came to Australia and he wanted to do animal trials, but I think he had more in mind doing rats, but he adapted pretty well to camels. So again, the feeding, the bleeding, the biopsies of the muscle biopsies. I will point out, camels tolerate muscle biopsies out of their rump a lot more than what they tolerate blood sampling. They hate it with a passion, the blood sampling. Muscle rump, you could take as much muscle out of their backside as they didn't seem too phased at all. So we did euthanasia and we did all the different tissues. And the animal ethics is up there. So all the tissues that were collected from the first 3 camels that were euthanized, indospicine was detected in all the analysed tissues at necropsy. So from muscle, there is differences between the tissues, but it doesn't concentrate in liver and kidney as a lot of toxins do. It's right through all the body, which is why it's so problematic when they're harvesting the meat. The pancreas was more than the liver and the muscle. But as I said, the differences weren't that huge. Then we did the depletion. So we fed all 6 camels up to this point. Then after stopping feeding, we just let them eat non-indospicine plant material for following 100 days. It took the depletion phase. It was still not 0 at the end of it, but you get to the point where animal ethics sort of overrides the return of data for what you're getting out of it. So we just stopped at that point. So the depletion half-life was calculated about 15.5 days in muscle, 18.5 days in plasma. That's how long it takes for it to drop by half. We also did some correlations between the muscle levels and the biopsy samples and the plasma samples, and showed that you can actually get a reasonable correlation, which is actually useful because it means that you don't have to biopsy so many samples. If you can get a blood sample, you can actually estimate what's in the muscle. We also analysed a whole lot of samples, both at the abattoir and field collected samples. So from the camel cull, we actually managed to get quite a few hundred samples that were actually culled in the field. So they were dropped by shooting from helicopters. But then there was other samples that were, the camels were transferred to the abattoir at Caboolture. There are only 2 camel abattoirs in Australia, one at Caboolture and one at Petersburg in South Australia. So out of our 197 ones that were killed in the desert, both in the Simpson Desert and Kings Canyon, there was ranges of samples. We looked at male, female, it didn't seem to make much difference. They were also divided into these groups of herds. Most of the levels that were high were in a couple of very specific herds. So it wasn't clear to us whether that was because there was just particular lots of Indigofera there or that particular herd just liked Indigofera. But it's certainly not all camels aren't bad, is probably a fair statement. We also looked at the abattoir samples. The abattoir, it takes a while to get from the Central Australia to Caboolture, and then even when they hit Caboolture, they are rested for a number of weeks before they're actually slaughtered. So in that time, it's almost like our depletion phase. So the levels, if there were any, were depleting. So the levels we saw at the ones at the abattoir are actually far less. Then we looked at, is there any way that you can actually treat the meat to get rid of the indospicine, which is what the industry were particularly interested in. Because the previous work suggested that indospicine, it was very much heat resistant. When Eddie did his trials, he actually looked at a range of pHs from the alkaline, the acid and the neutral. And we found that we could actually degrade it quite well with heating at the alkaline. So we got about 50% of the indospicine in the contaminated meat was degraded by autoclaving for 15 minutes in sodium bicarb. It does still seem a bit extreme, you're doing it for 15 minutes, but for a first trial, we were actually impressed that we could actually get it down that far and using something as mild as bicarb, whereas the water and the alkaline solutions remain the same. Again, the indospicine itself, when you actually use pure indospicine rather than the indospicine in meat, you get much the same thing, it just happens a lot quicker. So it's probable that the delay in the meat is just that it's taking a while to get it out of the meat. We did the identification of the products that came out of the hydrolysis. It's hydrolyzing the amidino and then going right through to the diacid. Pretty standard hydrolysis, but given that people said you couldn't hydrolyze it, we were actually impressed that we could actually identify the products. So this makes it possible to actually implement a similar treatment, maybe with some modification of the actual process, but if pet food was treated in this way, you could actually decrease your indospicine in it. And as any good rumen nutritionist says, amino acids are normally metabolized in the rumen of cattle and the foregut of camels. Why does indospicine escape the degradation? When we did in vivo studies with rumen fluid, we showed that you can degrade indospicine, but the degradation seems to be slower than the passage through the rumen or the foregut. So it looks like it's just bypassed. Basically, because it's a free amino acid, it's going through faster than we can get rid of it. However, Gabby's now looking at whether or not we can isolate the rumen microbes that actually can do the degradation. One thing we do know, there's that first step in the degradation within the human body, it just doesn't occur. There is no mammalian enzyme that's capable of degrading indospicine. But if we can isolate rumen bugs that can do it, that gives us potential that you can actually inoculate animals to protect them against it. So if you can degrade it in the rumen, it doesn't get into the meat and it doesn't flow onto the animal or the human food chain. So we're quite attached to our deuterated indospicine. It wasn't cheap. James didn't do it for nothing. Yeah, but regardless, if you actually look at how much time it saves and how much it's actually worth it to us. Because you can actually do so much more repetitive analysis, it has actually helped us get a long way with the studies that we've done. So just a couple of rounding up sides. The high levels of indospicine, not all Indigoferas are bad, only 2 of the 15 species, but unfortunately the 2 that, one of which has a large predominance, highest in new growth. I've shown that we've demonstrated the accumulation, persistent residues, half-lives of more than 15 days in both muscle and plasma. Again, we can see it in the camel meat samples that come to the abattoir and also the ones in the field. Generally, our levels were actually lower than what you'd expect toxicity with. So really not all camels are bad. And we are showing promise in that we can degrade the indospicine both in the meat and in the solution, and also hopefully in ruminal fluid. And sorry, natural is not always good for you. That's anyone who works in our lab always comes to that conclusion eventually. So I can't finish without thanking all the people who, there was a large contingent from DAF from when I worked back in 2003. They all seem to retire. It's, and Eddie who showed his enthusiasm throughout the project, despite the grueling hours we had him collecting plant material and feeding camels. So, and all the rest of our team down there. So, thank you.